Hey there, I'm Tyler Nickerson, and I'm here to talk to you today about cloning. No, not that kind of cloning. No, not that kind of cloning either. I'm here to talk to you about genetic cloning. I hate to break your bubble, but scientists today haven't yet invented technologies that allow us to clone dinosaurs or make instantaneous clones of humans. Sorry. That doesn't mean cloning isn't possible though. Scientists today use artificial embryo cloning and somatic nuclear transfer, also known as SCNT, to make clones of other living organisms. You see, sometimes when a woman is pregnant, the fertilized egg, called a zygote, tries to divide into a two-cell embryo. Because the two cells came from the same zygote, they contain identical genetic information. The two cells then continue to develop by themselves, and two identical twins will eventually be formed. But that's the old way of cloning. This is the 21st century. Since then, modern science has been able to make substantial achievements in the field of genetic cloning. In this video, we'll be visiting the far reaches of genetic engineering and find out what it truly means to clone another living organism. I'm Tyler Nickerson, and you're watching the step-by-step guide to successful cloning. The two main steps to successful cloning are learning cloning techniques and learning how to use those techniques to benefit scientific discovery. Let us begin by examining the two types of genetic cloning. The first type of cloning is the most simplistic. This form of cloning is called artificial embryo cloning. Imagine this ball of clay as a human zygote. Now imagine this cup as a petri dish. Once scientists have the zygote in view, they then will slice it down the middle, forming two new identical cells. Over time, the two cells will develop into two identical offspring. The second form of cloning, called somatic cell nuclear transfer, occurs like this. Scientists first gather an egg cell from a donor and a somatic cell, also known as a body cell, from the animal they wish to clone. In this example, we will be using a chicken. Scientists then extract the nucleus from the egg cell and dispose of it. Next, they extract the nucleus of the somatic cell and implant it into the egg cell. The egg cell then adjusts to its new nucleus. The egg cell continues to develop and eventually a clone of the somatic donor will be born. Okay, great, you can now successfully clone an animal. But why? How would that ever help you? Well, let's start by looking at a brief history of cloning. In 1952, scientists Robert Briggs and Thomas J. King cloned their very first animal, northern leopard frog. Over the years, more and more clones were produced, until in 1997 when scientists made a huge breakthrough in genetic engineering. Dolly the sheep, the world's first cloned mammal using an adult cell, was born. Dolly was created using SCNT and was the first success out of 277 failed attempts. She lived for about 6.5 years in a research facility until her death in 2003. 2 years prior, in 2001, scientists believed that the recent breakthroughs in cloning could repopulate endangered species. They cloned an endangered ox called a gar. Baby ox died only 48 hours after birth. However, scientists in Italy also cloned another endangered species, a sheep called a mouflon. The mouflon, named Embryo, is reported to still be alive to this day. Cloning is a great risk. Many of the animals produced die during or shortly after birth. Even if they do manage to survive, their lifespan is normally very limited. These defects can sometimes cause scare among consumers. Many consumers are concerned that consuming products produced from cloned animals can be detrimental to their health. Although markets today don't currently sell cloned products, there is still much concern that in the near future markets will begin to sell products produced from cloned animals. Now that you have a brief knowledge of genetic cloning and its history, may you go forth and contribute to the field of genetic engineering. Let's review what we learned. We learned that scientists currently clone organisms using two techniques: artificial embryo cloning and somatic cell nuclear transfer. Artificial embryo cloning involves manually dividing the zygote into two identical cells. SCNT involves transferring the nucleus of a somatic cell to the egg cell of a donor. Over the years, many scientists cloned various animals. Dolly the sheep was the first animal cloned using an adult cell. Scientists also attempted to reproduce endangered species. However, many of the cloned animals died at a young age or developed disease. Even Dolly developed lung cancer and only lived for 6.5 years. The reported defects of cloned animals scare consumers, and many are afraid that consuming products produced from cloned animals is detrimental to their health. Fortunately, markets do not currently sell cloned products. However, advancements in cloning are still being made to this day. Well, that's our show. Say bye, everyone.
Bye.
Bye. I'm Tyler Nickerson, and this has been the step-by-step guide to genetic cloning. See you next time.